Good morning, students. Today we are going to discuss about genetic diseases, and we know that Mendel, according to Mendel law of inheritance, there are three laws: law of dominance, law of segregation, law of independent assortment. And Sutton and Boveri correlated, and a linkage. There is a link between the Mendelian inheritance and the chromosomal theory of inheritance. And after that, linkage came into light and it hampers the crossing over. So linked genes always move together and express uh, accordingly. So it blocked the crossing over normal phenomenon, and then it came into light that there are some diseases that also carried or inherited from generation to generation from its parents to its offspring. So in this class today we are going to discuss about genetic diseases occurs in human being. So, how inheritance occurs in human? Since human and is in most of the cases 80 or 85, and even it crosses the 100 years, so it is quite difficult to study the human diseases directly and to study the several generation of a particular gene, whether it carries the disease inheritance gene or not. So in this case, a specific method is followed that is called pedigree analysis or study of the family tree. So in case of pedigree method, some systematic symbols are used to denote the affected and normal genes that carried by an individual. First, if it is rectangle, it denotes the normal male. If it is circle, it denotes the normal female. If it is square, then the sex cannot be identified. And if all of the three symbols or shape are red color, that means it indicates the particular individual is affected by disease, whether it is male, female, or unidentified. And if there is a link between rectangle and circle, then it denotes the mating or marriage involved. And if there is a two parallel line that connect the rectangle and circle, it denotes the marriage occurs in or between the two persons, male and female. They are quite related with each other. And in next case, it denotes that the offspring or the child produced after marriage of an individual, that indicates they are both normal and there is no expression of any kind of genetic disorders. But in some cases, the father and mother both are normal. The particular character cannot be expressed or they are unable to express, but they sometimes act as a carrier, and in the next generation they express in male offspring. And in the last symbol, the square shows the 5 out of the 5 unidentified sex or offsprings are unaffected. So these are the symbols used in pedigree analysis to denote the particular kinds of disease. So why genetic disease occurs? The genetic disease occurs due to a particular phenomenon that is called mutation. We know that mutation is a sudden inheritable changes that occurs in DNA because it changes the sequence of gene in DNA, and it is called mutation. And the organism that undergoes mutation is called mutant, and there are various kinds of chemicals or environmental factors also that are the main cause of the mutant or mutation. So those as chemicals that helps in causing mutation are called mutagens. And in mutation, actually it alters the DNA sequence that results in the expression in phenotype and as well as in genotype of an individual. So there are three main specific points regarding mutation. First one, due to the loss or deletion or gain or insertion or duplication of a segment of DNA, that as a result, the alternation of chromosomal sequence or chromosomal abnormalities occurs, and these types of mutation is called chromosomal aberration. In second case, sometimes gene mutation occurs. Gene mutation takes place at a particular place due to the changes in a single base pair in the DNA. It is called gene mutation or point mutation. And in the third case, sometimes deletion and insertion of a pair of gene occurs in DNA. Then it is called frame shift mutation. So these are the three different types of mutation, as a result which mutation occurs in an organism. In our next slide, we are going to study about the different types of genetic diseases. So basically, there are two types of genetic disease. One is Mendelian disorders, other is chromosomal disorders. So in today's class, we are going to discuss about Mendelian disorders. And Mendelian disorders are mainly determined by alternation of mutation in the single gene. That means it causes the change in the single gene pair or the single DNA gene part. It obeys the principles of Mendelian inheritance, but during transmission from one generation to next generation, the disease expresses that actually due to the gene or point mutation. So these kind of genetical or Mendelian disorders, for example, can be traced with the help of pedigree analysis. For example, haemophilia, color blindness, cystic fibrosis, sickle cell anemia, phenylketoneuria, thalassemia, etc. The genes can be dominant or recessive, that also can be determined with the help of pedigree analysis. And the traits either may be sex linked, that is X chromosome recessive linked, or it may be autosomal linked genes. For example, sex linked inheritance of Mendelian disorders is haemophilia. And again, sex linked recessive traits always transfer from carrier mother to its son, and from son to its daughter in the next generation. So the transmission always occurs in rice cross method. Next, today we are going to discuss only Mendelian disorders. So in this class, we are going to discuss haemophilia, sickle cell anemia, and phenylketoneuria. So what is haemophilia? It is a sex-linked recessive disease. The defective individual continuously bleed even in a single cut. That means sometimes a simple cut and a very short cut may occur in an individual, but it causes excessive bleeding and leads to the death of the patient. So it is also called bleeder's disease or royal disease, as initially it was found in royal family of England. In this disease, the single protein that take part of a cascade of protein that involve in the clotting of blood is mainly or basically affected. The disease transmitted from unaffected carrier female to its male progeny or to its son. So heterozygous female always act as a carrier and transmit to its son in a criss-cross manner, while female being hemophilic is very rare in this, but in this case they must be homozygous. That means both the X chromosome must carry the hemophilic gene. So in this picture, we are going to determine the Punnett square chart. So here, in the first trait seen, here the father is affected by hemophilia, mother is normal, but in the next generation, the father transmit the disease to its daughter, so it appears in the daughter in form of carrier, since it is found only in one X chromosome. It is heterozygous condition, so it acts as a female carrier, but the son is unaffected and normal. In the next case, here the mother is carrier, father is normal. Then in this case, the one son and one daughter is always healthy, but the one son that is affected by hemophilia and one daughter that is always act as a carrier. So these are two different probabilities. In the first case, the father is hemophilic, and in the second case, mother is carrier, and the probabilities are as given in the chart. Next disease, we are going to discuss about sickle cell anemia. Sickle cell means the RBC shape always biconcave, but due to this defect, the shape of the RBC changes into sickle-like. Why it occurs? Because it is due to the changes in the genetic code in the 6th position of DNA that transfer to RNA in form of coded information. That means in DNA it is GAG, but in mRNA it transferred in form of GUG. So instead of glutamic acid at that position, the protein and polypeptide chain and replaces the glutamic acid to halide at 6th position. So in the next picture, you see the normal HB gene, hemoglobin gene A. It always denotes GAG in DNA. When it transferred to mRNA encoded information to carry the amino acid in and form of protein in ribosome. Here, mRNA has coded information GAG for glutamic acid as at 6th position of HbA polypeptide chain. But in case of sickle cell anemia, here mRNA carry the GUG at 6th position of HbS polypeptide chain, that is defective, and so glutamic acid is replaced by valine at 6th position. So the shape of the RBC changes from biconcave to sickle type. So, what are the main cause? It is due to the genetic disorders, and it is an autosomal linked recessive trait. That means the gene in recessive form, not in dominant form, it is found in bodily carrying in chromosomes, not in the sex chromosome. So it transmits from both parents are carrier. Sometimes both parents act as a carrier, so it transmitted the disease, and the disease is controlled by single pair of alleles. As we know that for a particular character, there are two alleles present in the chromosomal locus, but here we found SBA and SBS. How it plays act as a heterozygous role. Next, in homozygous individual, the disease appears. That means in here, in case of some individual SBS, SBS, then the individual is diseased. In heterozygous individual, it is not affected, but it acts as a carrier. That means in homozygous condition, the disease is expressed either in male and female, but in heterozygous condition, it acts as a carrier. Why it occurs? Because due to the substitution of glutamic acid by valine at 6th position of beta globin chain of hemoglobin. So due to this substitution, the code, what is the main code involved? At 6th position, it must be GAG, so that glutamic acid can sit. But in case of sickle cell anemic patient, at 6th position of polypeptide chain, GUG sits, so or it denotes the valine instead of glutamic acid. So the shape changes, and as a result, it cannot carry oxygen, and it causes defect in the patient, and oxygen deficiency may lead to its death of the patient. So here is the different probabilities how the disease transmitted to its progeny or to its offspring. In the first case, the father is carrier, mother is normal. Then in the next generation, two of the child are carrier and two of the daughter are normal. In the second case, when the father is sickle cell anemic, mother is normal, then in the next generation, all of their childs are act as a carrier. And in the third case, when both of them are mother and father are sickle cell anemic, then in the next generation, all of their childs were sickle cell anemic. In fourth case, when both the parents are carrier, then in the next generation, one is sickle cell anemic, one is healthy, and other two sons and daughters are act as a carrier. And here you see when the father is hemophilic, mother is carrier, then in this case, two of the child of them are act as a carrier, and two of the child of them are sickle cell anemic. So there are different probabilities depending on the carrier and whether the mother carry the genes or the father carry the genes. The probabilities are such types. In the next, we are going to discuss about third genetic disorders, Mendelian disorders, that is called phenylketoneuria. It is an inborn error of metabolism. That means it affect the metabolism of an individual. And here, the gene is actually found in autosome and in recessive form. The affected individual lack an enzyme, and that enzyme is very essential to convert amino acid phenylalanine to tyrosine. The phenylalanine accumulate and convert to phenylpyruvic acid and other derivatives, and it accumulate in brain, results mental retardation, and it also causes excessive urination that releases excess water from its body and causes dehydration. So in the next table, you see the enzyme that is responsible for phenylketonuria, the lack of enzyme that is called PAH enzyme, and as a result, toxic amount of phenylalanine accumulates that converted into phenylpyruvic acid, and it causes atrophy and mental retardation. So today in this class, we have discussed about Mendelian disorders. First is haemophilia, second is sickle cell anemia, and third is phenylketonuria. Thank you, students, for attending the class.